Hey everybody, and welcome to the first lecture for MLT 140 Clinical Microbiology. In this first unit, we're going to be covering some different topics, including taxonomy and morphology, getting into some parts and functions of different types of microscopes. We're also going to be talking about the first group of bacteria that we will discuss in this class, the Staphylococcaceae. So this Unit 1 lecture will be broken up into a few different parts, so just make sure you watch all of the lecture videos. So the first thing that we'll talk about are some of the different branches of microbiology. So bacteriology is the study of bacteria. The bacteria are unicellular organisms. They lack a nuclear membrane and a nucleus, so they're referred to as prokaryotes. Mycology is the study of fungi. Fungi are organisms that obtain nutrients through absorption, so yeasts are a type of fungi. Parasitology is the study of parasites. Parasites gain nutrients from a host. Virology is the study of viruses. Viruses are the smallest infectious particles. They cannot be seen with an ordinary light microscope. They are not living cells, so they require a host for replication because they cannot reproduce on their own. And then epidemiology is the branch of medicine dealing with the incidence and prevalence of disease in large populations. They also deal with detection of the source and cause of epidemics of infectious disease. So how would this apply in a hospital setting? 5 to 6% of hospital patients develop nosocomial infections. So those are infections that the patient develops while they are in the hospital. The UTI, or urinary tract infection, is the most common type of nosocomial infection. And this is due to patients having catheters, improper hygiene techniques. Most common cause of a UTI is the bacterium E. coli. And infection control personnel within the hospital review lab results daily in order to track the spread of infection. So when we have patients come into the hospital, we don't want them to get sicker while they're in the hospital, so we need to make sure that we're practicing sterile processes and infection prevention. So hospitals are going to have folks on staff that track those things. Going into a little bit of terminology now, just kind of an overview of some terms. Normal flora, which is also sometimes called indigenous flora, is the normal bacteria that we have in our bodies that does not hurt us. So different body sites have different normal flora depending on the nutritional requirements of the organism. So does it need a moist environment? Does it need a dry environment? Does it require a certain pH? The 4 general sites of normal flora in the human body are the upper respiratory tract or the mouth, genitourinary tract, including the vagina, skin, and the GI tract or the intestines. A pathogen is a microorganism that causes disease. You can have what are known as true pathogens or opportunistic pathogens. So true pathogens are organisms that are recognized to cause disease in healthy, immunocompetent individuals. So an example of that would be the bacterium Yersinia pestis, which is the organism that causes the plague, and Bacillus anthracis, which is the organism that causes anthrax. So even in a healthy person, if that person is exposed to these organisms, they will likely get sick. An opportunistic pathogen is normally non-pathogenic organisms that can cause disease if they are given the opportunity to do so. So immunocompromised patients, which could be infants or the elderly, may get sick if exposed to an organism that a normal healthy person would not get sick if exposed to. An example of this one could be the bacterium Haemophilus influenza. A commensal is more of a neutral organism, so it gains benefit from the host, but the host is not hurt by it. example is Proteus mirabilis in the GI tract of humans. So it just kind of hangs out there. It's meeting its own nutritional requirements without hurting the host. A nosocomial infection, infections that are acquired within the hospital setting within 72 hours of beginning their hospital stay. And again, we have folks in the hospital that track those types of infections. Iatrogenic infections are those that are the direct result of a medical treatment or procedure. So an example would be a UTI caused by an indwelling catheter. And then virulence is the relative ability of a microorganism to cause disease or the degree of pathogenicity. So this is usually measured by the number of organisms that are necessary to cause infection in the host. An example of this would be the organism Shigella, which is considered to be highly virulent because it requires such a low infective dose. So it requires only around 100 organisms if a patient is exposed to actually cause disease. Next, we're going to talk a little bit about the actual microbiology laboratory. So some safety considerations. Aseptic techniques are practiced in the micro lab. So aseptic means without sickness. So the goal is to reduce microbes in a medical setting and at preventing wound infections. So we do this by hand washing. And this is another thing that it's considered so important that hospitals track this among personnel. They have kind of secret shoppers that will go around the hospital and watch hospital personnel to see if they are washing their hands, either when they enter or when they come out of a patient's room. And we do all this by also do this by avoiding aerosols. So sprays produced by the buildup of heat or gases. So you have to be careful when opening some specimens. We do this with a laminar flow hood or biological safety cabinet. They are identified as either class 1, class 2, or class 3, depending on the types of organisms routinely handled and the hazard that these organisms pose to lab personnel. So the organisms themselves are classified as biosafety levels 1 through 4. Most labs are going to have a class 2 hood. These pull the air up and away from the tech. They have a HEPA filter, so it helps get rid of different pathogens and keep those away from the lab tech so that they're not breathing in these aerosols. Disinfection is a process that eliminates a defined scope of microorganisms, including some spores. There are physical or chemical methods that can be used for disinfection. Most are chemical agents applied to inanimate objects. So some methods of disinfection. Chemical methods would be bleach, and a 1 to 10 solution is commonly used in the lab. Iodine, alcohols, aldehydes, hydrogen peroxide. And then some physical methods to include boiling. So you can boil at 100 degrees Celsius for 10 minutes. 10 minutes, that would kill most organisms. Pasteurization is boiling at different degrees for varying lengths of time depending on the substance, and that's used mainly for food preparation. And then using non-ionizing radiation such as UV rays, non-effective because it has to have direct surface exposure. And then antiseptics are a disinfectant applied to the skin or tissue to kill or reduce the number of organisms, so to prevent infection. It won't kill spores, cannot be used as disinfectants, and these are used for personal lab safety and can also be used to prepare patients for blood draws or other procedures. Sterilization is a process that completely removes or destroys all forms of microbial life, including spores. There are no degrees of sterilization, it's all or nothing, and this uses mainly physical methods. So some of the methods of sterilization include incineration, which is a method of treating infectious waste where it is burned to ashes at temperatures from 870 to 980 degrees Celsius. It's the safest method to use to ensure that no infective material is left, but toxic air emissions and heavy metals in ash can limit its use in the United States. You also have moist heat, which is also referred to as autoclaving. An autoclave is used, and it's essentially just a big pressure cooker. So it's used to sterilize biohazardous trash and heat stable objects. 15 pounds of pressure for a minimum of 15 minutes at 121 degrees Celsius for broths and solid media and instruments. Infectious waste is usually going to be at 132 degrees Celsius for 30 to 60 minutes, and it's faster than dry heat. Dry heat is commonly used for glassware, oils, or powders, but it can cause harmful emissions. And then filtration can be used to filter liquids such as antibiotic solutions, toxic chemicals, vaccines, liquids. Use a thin membrane that the liquid is pushed or pulled through. And then you've got air filters called HEPA filters that are used to clean the air. Some are even small enough to filter out viruses. And then radiation is used by medical field for sterilizing disposable supplies such as syringes, catheters, or gloves. And then universal precautions. So these are guidelines stating that blood and body fluids from all patients must be treated as infectious and capable of transmitting disease. So one of the ways that we practice universal precautions is by using our PPE with all samples and with all patients. Now getting into some bacteriological equipment. So a medium, or media for plural, is a nutrient used to grow organisms outside of their natural habitat, and it can be solid or liquid. A lot of times you might think of it as a petri dish. Maybe you've seen pictures of a petri dish before. An incubator is defined as an apparatus in which media inoculated with microorganisms are cultivated at a constant temperature. Different organisms are going to require different temperatures for optimal growth, but the most common is 36 degrees Celsius, which is body temperature. A loop, or a calibrated loop, is a device used to transfer organisms from one medium to another. They can be plastic or metal. And then an incinerator is an apparatus for burning waste or used to clean metal loops before reuse. So most hospital laboratories now are going to use disposable loops. If you do use metal loops, you have to incinerate them before you reuse between organisms. And finally, in this part 1 video for the Unit 1 lecture, we're going to cover some parts of the microscope. So the ocular lens is the eyepieces that you're going to look through on the microscope. So right here, the magnification of the ocular is always 10x. Then the objective lenses, that would be these here, are the lens closest to the specimen. They're going to come in different magnifications, so it can be 10x, which is considered low dry, 40x, which is considered high dry, or 100x, which is our oil immersion. There are other magnification or other objectives, but those are the most common. The nose piece is the piece that holds the objectives that revolves, so you can switch between your objectives. The stage is the surface where you lay your slide. The stage control moves the stage so that different areas of the slide can be viewed, so you can move it forward and backward and side to side. The base is the bottom weighted part of the scope that sits on the table. The light source or illumination is controlled by a rheostat, so it controls the height of the light and the amount of light coming through. The rheostat is a way to adjust the brightness, so it regulates the light intensity. It's controlled by a dimmer slide that rotates right to left to control the light. The condenser is between the light source and the slide, so it focuses light on the specimen. And then the fine and coarse adjustment knobs are for focusing different objectives. So usually when you're initially trying to focus on a slide, you're going to start out on the lower objective, so the 10x objective, with the coarse adjustment to get it focused initially, and then you can move up to the higher objective and use your course adjustment to fine-tune your focus. Some terminology related to the microscope. Resolution is the extent to which detail in the magnified objective is maintained. So without it, everything would be magnified as an indistinguishable blur. Resolving power is the closest distance between two objectives or objects that, when magnified, still allows the ability of the microscope to delineate between two adjacent objects. Parfocal is the ability to move between two different objectives and maintain focus. So if you start focusing on your 10x, you get it in focus there, and then you move up to your 40x objective. Parfocal is the term related to how that object will stay in focus between the objectives. Definition or contrast is the ability of the scope to clearly define objects so that they stand out from the background. And then to determine total magnification, you have to take the ocular, which remember is always 10x, and multiply it times the objective that you're using to view the slide. So if, for instance, if you're viewing the slide on the 40x, the total magnification would be 10 from the ocular times 40 from the objective to be a 400x total magnification. So why do we use the microscope in the clinical lab? In the microbiology lab, the main objective is for rapid preliminary organism identification in specimens. So we do this with what's called a gram stain, which we will get much more into later, but it's a type of stain that allows us to categorize bacteria based on cell wall features, but it can give a preliminary identification to what the organism may be. Wet prep is when we take a direct smear of a body fluid and add a drop of saline. So we, for instance, if a patient had a wound, we could take a swab and get a sample directly from that wound, wipe it on the microscopic slide, add a drop of saline, and then place a cover slip on top so there's no stain performed. Then we can examine that under the microscope. So this is commonly used for vaginal exams to look for yeast, certain bacteria, and what are known as clue cells. And then AFB stain or acid-fast bacillus, this is the type of organism that causes tuberculosis. So all three of those would be means of rapid preliminary identification that could help the doctor initiate treatment. It could help with detection of different organisms present in the same specimen, which could be an indication of possible contamination. So it can give us an idea to the quality of our specimen. Detection of organisms not easily cultivated in the laboratory. So if it's one that is hard to grow on media, we may still be able to see it on the gram stain or a wet examination. It helps us with evaluation of patient specimens for the presence of cells indicative of inflammation or infection versus contamination. So again, just kind of a quality check on our specimen. And then it can help provide pre-culture information about which organisms might be expected to grow so that treatment can begin if necessary and to correlate with culture findings. So our gram stain or our wet prep results are usually out within, I would say, 30 minutes of us receiving the specimen in the lab, whereas the culture, which is the growth on the media, has to incubate. It usually takes about 16 hours, give or take, before we can give a preliminary result from the culture. So those stains and wet preps can provide initial, very quick information as to what type of organism might be growing and if it is a true infection. It also helps us predict what should be growing on the culture. So if we report out an organism from the gram stain, but then the next day when we're reviewing the plates, there is no growth on the plates, then that does not correlate. So we would have to do some investigation into why that is. In this last little bit of the Unit 1, Part 1 lecture, we're going to go over some different types of microscopes. So the bright field microscope is the most widely used type of light microscope. That's what you will use in our classroom laboratory and most commonly in the hospital laboratory. The background appears light, the objects are going to appear dark. With phase contrast, this allows for viewing of clear objects in a liquid or unstained cells by the way light is bent around the objects. This is done with a special condenser that's added to a bright field microscope that changes light waves passing through the specimen into differences in light intensity. So with this one, the background will appear gray and the specimen appears bright. With a dark field, this is similar to phase, so you've got a bright field microscope that's been modified with a disc filter. The background appears dark, the objects are light because the light is blocked so that only reflected light is hitting the object. We use these to view live cells that would be distorted by drying or that can't be stained by the usual methods. And then the electron microscope, there's a couple different kinds. They're rare, they're not commonly used. They offer extreme magnification, and they're going to provide details of structures of cells and viruses. Lastly, just make sure that you take a look at the actual PowerPoint file in Moodle, as there are a few review slides after the end of this lecture. You could use those review slides to just help test your comprehension or recall of some of the topics that I just talked about.